So I'd like to start by really thanking the organizers, Dr. Grafe and Dr. Heckenreuther, for organizing this symposium and giving me the chance to tell you about a bit about our activities in micro NMR. I will acknowledge co-workers individually at the end, but I like to put that slide up first because I'd like to be reminded that science is a collective effort, and of course what I'm going to show you is not my work, it's mostly the work of people represented here. So let me indulge me in allowing me to take a bit of a step back and philosophize a little bit. So if we look at the grand challenges that are facing humankind at this point, if I asked the audience for suggestions of what to put on this list, I'm pretty sure you would all come up with some permutation of these issues here and maybe a few others. And after a bit of thought, one could probably find that they all have something in common. If we want to address them, they have a biological side to them that's more prominent in some cases and maybe a bit less in others. But in order to really tackle these issues into the future, we will have to find ways to derive quantitative biological insight from biological systems. What do I mean by a biological system? Well, I mean essentially almost about anything. It could be a system at a large scale. It could be an entire ecosystem of many organisms working together. But also, of course, each individual organism is a biological system made up of smaller systems, made up of cells, made up of organelles, made up of molecules. And of course, a biological system could be defined at any length, at any point in this continuous scale. So this is, of course, unscientifically generic, what I'm telling you here. So let's try and find the red thread that runs through all of this. At any scale, biological systems are always part of an environment that they function within. They're always complex, and they're always diverse. That's two different kinds of complexity, you might say. That you have copies of, or individuals that are all slightly different from each other, and all of them, each of them, is of a highly complex nature. So my point is that lab-on-a-chip systems, which are a very rapidly developing field of science and technology at this point, and I think at this conference you will find many examples of such systems, address these three characteristics almost directly. So the fact that a biological system is a part of an environment can be mapped in a microfluidic device. For example, here in this chip here, this is a PDMS chip that mimics the functioning of the lung by adhering endothelial and epithelial lung cells onto a membrane, and that membrane can then be stretched by simply evacuating the channels here on the two sides. So you can create the environment and control it. The other aspect is that you need, in order to deal with the complexity, you need to get data-rich analysis out of it, and that could be done by various different approaches. In this particular case here, this is a study of primary hepatocytes, and there is a broadband immunostaining approach that's used here in order to follow the differentiation or de-differentiation of these cells. And there are high throughput techniques that are developed in this field of lab-on-a-chip systems that allow dealing with the diversity of biological systems. In this case here, this is a microscopy assay that uses a laser to evaporate essentially small samples of the microscope image, and you can actually select in the microscope which part of the sample you want to zap in this way. And then there is a small an array of small droplets of DMSO that pick up the debris, which is then fed into a mass spectrometer. So you get a spatially resolved at the individual cell level answer on the proteome of such a sample. So now, of course, this is an NMR session. Why do I think NMR has a role to play in all of this? NMR is a systemic technique, so it gives an answer or a response if you put a biological system in there. We've already seen beautiful examples of this earlier today. You get essentially everything. You see potentially every metabolite. So in that sense, it's a generic technique. It can be quantitative if you do it right. You can integrate peak intensities, and they give you direct concentrations. It can be non-invasive. So you can do this and the sample lives on. I've also seen a beautiful example of that earlier, and of course that implies that it's non-destructive. So in what we have in mind, we would like to do the following. So a typical lab-on-a-chip type model systems are operated in a timeline that looks like this. You start by seeding some biological system, maybe some cells or a tissue slice, onto the device, and then you culture it for a certain period of time. And during that time of culture, of course, one can add stimuli, for example, feeding a drug or changing the flow of nutrients or some other influence that we would like to study. And then you could even do this multiple times, and at the end, one typically destructively tests the system by some sort of very often fluorescent-based analysis or mass spec-based analysis. These workflows are very well established, they're highly sensitive, and I'm not proposing that we replace this arsenal here with NMR, of course, but I am arguing that by taking NMR spectra in this phase here, we gain additional information on the metabolic history of the system, which we can then hopefully correlate with what's happening here. So here is an example of how we are trying to do this. This is just a demonstration of principle where we have grown MCF7 cells in a small microfluidic device in about 2.5 microliter sample chambers, and we did it in slightly different ways. In one case, we coated the inside of the chip with pluronic. That's a polyethylene glycol type material that makes it very difficult for the cells to adhere. And since these types of cells that we use don't like to be non-adherent, they adhere to each other and they form these spheroids. And in the other case, we made it easy for the cells to adhere by using fibronectin, and you get these two-dimensional, more conventional cell cultures. And we periodically stuck these microfluidic chips into an NMR spectrometer. I'll tell you more about the technical details in a moment. And we can get NMR spectra each time from these 2.5 microliter sample volumes that look like this here. We can then go through the human metabolome database and essentially solve the inverse problem of how much of each of these metabolites do we have to add together in order to create the closest possible fit to the experimental spectrum. That gives us compositions, which we can then now plot over time, and we can extract metabolic rates for these two different experiments. And one finds that the rates are quite different depending on whether the cells are spheroids or adherent, which is not surprising, of course. But for us, the interesting bit was, can we quantify this? What are the signal-to-noise ratios? What are the sensitivities? What are the error bars? And so on. So that's that's why we did this. That's what was our motivation for this work. So this is based on a strip-line type probe, which you see here. This is an NMR probe that just replaces the standard 5 millimeter probe that you may have in your spectrometer. And it's based on a strip-line geometry here, which is a slight variation of the geometry that has been pioneered by the group of Arno Kenkens, in that we use two of these strip-line geometries. We put them with a spacer next to each other. So the sample then is in a microfluidic chip, which is designed to have its sample chamber aligned with the constriction, and that gives us high sensitivity. We did some topology optimization to find the right shape for the strip line and the sample chamber simultaneously, and that gave us the best compromise between homogeneity of the B1 field and probe efficiency. And that then allowed us to get these spectra. I'll talk about sensitivity a little bit later. So more recently, we've become more adventurous, and we've started to look at tissue slice samples. So this is a mouse liver here from which we core out cylindrical samples of about 5 millimeters diameter, which are then microtomed, and you end up with tissue slices that are about 2 to 300 microns thick, and the same, of course, diameter as the core here. These samples can then be cultured. Conventionally, they're put in a well plate, but we can culture them in a microfluidic device that has a culture chamber up here, and it is connected by a fluidic path to the NMR sample chamber. We supply the tissue with nutrients by a syringe pump, which flows the nutrients past the tissue and then into the sample chamber where it gets analyzed. And of course, to keep tissue alive, rather than cancer cell lines, you have to be careful about oxygen supply. So we supply the oxygen to this system through this blue gas channel, and the red and the blue channels are both covered by a PDMS membrane so that the gas diffuses through the ceiling, as it were, from one channel into the other. So this allows us to get continuous in situ or in operando spectra during the culture. That you see some samples here. In this case, we switched out the gas supply halfway through the experiment. We essentially asphyxiated the tissue at a certain point in time, and you can see that here almost immediately there are changes in the NMR spectra, and of course the first thing that reacts is the herpes buffer that we had in the media feed, because of course immediately the tissue goes into acidosis, and the carbonate buffering no longer works, and you can see that here. There are also more subtle changes in other signals that I'll talk about in just a moment. One point I should mention is that we are interested in measuring changes. We want to see metabolic rates. We're not particularly interested in absolute concentrations. And if you want to see change, well, then you need to allow time for it to occur. And that leads to a problem here, because in order to provide the tissue with sufficient oxygen, you have to maintain a minimum flow rate. If you go below that flow rate, the tissue consumes oxygen more rapidly than you supply it. After all, we're supplying it through an aqueous solution, which is terrible. Right? That's why we have blood, because oxygen solubility in water is terrible. So you need to maintain a high flow rate. If you have a high flow rate, everything that happens in the tissue gets diluted into a massive amount of perfusion fluid, and you never see any difference. You always measure the same concentrations for everything. So the way we solve this is we use a back and forth pumping approach, where we take the same plug of fluid of about 20 microliters that goes past the tissue and then comes back. We just reverse the flow. And each time it goes past the tissue, each time it goes past this part of the structure, it gets reoxygenated so that it continues to be used. And this is reflected in these concentration curves here. So each time when you see an arrow here in the timeline is when we refresh the medium. So we just let the syringe pump run a little longer on the in-stroke so that what we used before gets pushed out and replaced with fresh medium. And you see here that each time then the concentration, for example, of lactic acid goes back to nominal, the concentration that in the feed. And then you see the tissue producing some more, and then it goes back to the feed concentration as well. And you also see that the rate at which the tissue is doing this decreases over time, and that of course has to do with the decreasing viability of the tissue in this environment. So the goal of optimizing this, of course, would be to make sure that these decreases become less rapid, that you can maintain the tissue viable for longer. Now, the data-rich aspect of this is that we can get concentrations of a large, well, I hesitate now because I was in a mass spec talk earlier this morning, and the mass spec people wouldn't wouldn't call this a large different variety of metabolites because they look at hundreds of them. But for NMR, this is pretty respectable. We can see 20 to 30 different metabolites, and we can quantify them in these 2.5 microliter sample volumes. You can now, of course, try to make sense of this data, for example, by fitting each of the individual plugs with a rate equation, and you get information on the metabolic activity in the tissue. Some of them are totally obvious, right? It's making massive amounts of lactic acid, some acetic acid. If you were in a beer garden last night, that's a process that might be happening in your liver as well. And it consumes a bit of glucose. We've actually seen some samples produce glucose, which seems counterintuitive, but it's just a glycogen decomposition that you see in liver tissue, of course. So here are some examples enlarged, and the one I found particularly amazing was this one here, L-tyrosine. That's present at a concentration that we can barely see in the spectra. It's really in the noise. However, because we take so many spectra and we do then a fitting of these signals, we can actually see that there's a trend in this noise that is statistically significant. Otherwise, it wouldn't always be a positive slope every time we measure it. So we actually do see tyrosine production in these samples here, and we can quantify it at about 10 micromolar per hour, and that's about our limit of detection with this methodology. We've pushed this a little further and compared the metabolic response of liver tissue to a cholestatic drug, and we can see clear differences for a number of metabolites, not all of them, where the production rates of or consumption rates of lactate and glutamine are quite different. We can even do imaging. So in order to do that, of course, we have to put the tissue slice where the NMR detector is, and we get images that have resolution that is sufficient to see the microanatomy of the liver tissue. So you see a tiny portal vein here, and if you squint, you can actually see some similarity between this cartoon and what we're actually seeing in the tissue here. So let me shift gear for a moment and talk about automation. So microfluidics, of course, is good at looking at implementing entire lab procedures on a chip, and we've used this to build a system that can do titrations in situ. So in order to do that, we've incorporated some valves up here in the chip, which are pneumatically activated, and that allows us to inject a small amount of sample, much less than the probe volume, and then mix it by circulating it around through peristaltic pumping, and then observe the NMR spectrum. And we've used this to do an automated study of binding for a protein. We used a sample that's quite popular as a guinea pig in NMR. We used the SH3 domain, which binds to a proline-rich peptide in a process that's very well understood. And we can then get these two-dimensional NMR spectra out of our system. And you see some of the metal peaks shift here. And from the peak shift as a function of mixing ratio, we can get the binding constant. So this uses a minimal amount of solution to do this entire experiment, and you can do it in a completely automated way. You inject it once, set the experiment, set go, and collect the data the next morning. Now, let's talk briefly about sensitivity. We've heard a lot about this already, and it turns out that this double strip line geometry that we're using here is actually a very useful instructional paradigm to calculate the theoretical limits of sensitivity for inductively detected NMR. So imagine that you have a sample here, which contains a nuclear magnetization, which is precessing, so it's oscillating, and it's confined by two conductors here, and these two conductors then respond by an electromagnetic wave. So the electric field is plotted here. This is just a simulation of Maxwell's equations, and you see the oscillation here caused by the sample here in the middle part where this straight line oscillates, and there are two waves that are traveling to the right and to the left. And as I said, this is Maxwell's equations. This is maybe not first-year physics, but certainly third-year physics to calculate this. And we can calculate everything. We can calculate the magnitude of this field and specifically the amount of power that is coupled into these two traveling modes. At the same time, we can calculate the noise power that comes from the finite conductivity of this part of the transmission line. And then you compare signal power to noise power, and you get the signal-to-noise ratio with no adjustable fudge parameters or assumptions. And one can then simply calculate the scaling of this with size. What you find is that the sensitivity of such a detector, an ideal detector, will scale along this line here. You get a volume scaling of a power law with a slope of 1/3. And you see actually, if you look at data that has been published by others in the literature, that, yeah, well, if you squint, you can probably recognize some kind of slope like this in this crowd of points. Our transmission line probe is here, and the commercial cryo probe is this blue point here, and the very nice rolled up coils that were just explained to us so nicely before are, let me see, they're at a detection volume, they must be around here, somewhere here, right? And they are very nicely on the same line, I think. So now, if you want to beat sensitivity, if you're already at this line, then you have to do something extra. You have to increase the polarization of the nuclear spins. And there are various ways of doing this. The one we are interested in is uses parahydrogen, so uses a spin order in one of the allotropes of hydrogen gas in order to achieve a higher spin polarization. As the polarization at room temperature is only about 1 in 10 to the 5, potentially there are 5 orders of magnitude in sensitivity to be gained. Of course, in reality, what one actually achieves is less. So here is an example of a chip we use for this type of research. We use a very similar idea in order to get hydrogen gas into this solution that we used for oxygen before. And I don't want to bore you with the gory details. I'm very happy to talk to you after the session about the details. But we can get sensitivities which are now detecting picomole amounts of material, but of course it's now no longer generic. We can only detect the sensitivity of the hyperpolarized material. We are doing quite a bit to optimize this. And one of the advantages of doing this in continuous flow is that you can do two-dimensional hyperpolarized NMR. This is very difficult for other people who in this field, who do this in 5mm sample tubes that they pressurize with hydrogen and have to shake the tube outside of the magnet and then very quickly insert it into the magnet. If you have to do that for every increment of a 2D experiment, you never do it the same way. Well, you laugh, people have tried. You never do it the same way twice, and of course you get a lot of T1 noise, and we can just do it essentially in an experiment like this. So we take this quite seriously. We just got the grant to develop this idea further, so we try to kinetically model exactly what's going on in this device, because it's a very complicated coupled chemical reaction, transport, and of course nuclear spin problem that's happening. So it's actually quite fun to play with it, and we're getting better at actually simulating what we see in the experiments. So finally, we've managed to actually directly hyperpolarize a metabolite, and the idea, of course, is then to expose a biological system to the hyperpolarized metabolite and detect the downstream hyperpolarized metabolic products. So that we haven't achieved yet, but we have achieved hyperpolarization of fumarate in an aqueous solution. There are some intricacies to this because fumarate is a highly symmetric molecule, so one generates a singlet state here as opposed to a dipolar state. for those of you who are spin gymnasts. And of course, we would like to ultimately see the signal from this carbon-13 labeled system here. We would like to have the carbon-13 response because it's longer lived, but when we tried so far, we haven't been successful, and it turns out that that was because the hydrogen uptake in our chip wasn't good enough. We used the simulations to improve, to guide us to how we can make the chip better, and got a better hydrogen uptake at a higher flow rate, and that shifted the balance to enough so that we can now actually see, and this is really fresh data, this was collected last month, we can actually see the carbon-13 hyperpolarization in this microfluidic system as a function of flow rate here. So I'd like to conclude very generally, about as non-scientifically general as I started. I think there are a lot of opportunities for research and advancement in the combination of microfluidics and NMR. And I think that future developments are going to be driven by forces that are powerful and that are in motion elsewhere in science and technology. One is recent advances in manufacturing, additive manufacturing in particular, real breakthroughs. Automation is going to be a very big element, I think, in this story in the end, and a very important aspect to deal with the data richness of the stuff that comes out, also artifact removal, etc., is going to be integrating advances in data science with endeavors such as the ones I have told you about. So finally, let me acknowledge the team here. This is a pre-pandemic picture of my research group. Many of these people have now moved on to better things to do, but some of them are still with us. So Patra, here to start on the left, was a postdoc who's worked on the tissue slice culture. Then his sidekick was Manvendra on the other side. He's been building our probes and has been absolutely key to all of this. And I'd also like to point out Sylvia, who's been working on the hyperpolarization. Marek is a PhD student. He's in his last few months. He's been working on the protein NMR work. And of course, I'd like to acknowledge my colleagues in Southampton, particularly Malcolm Levitt and Jörn Werner, my colleagues Sabeth Verpoort and Jan Korvink, who were project partners on our EU Fed Open grant, and various institutions have lent us their financial support. And I'd like, last but not least, to thank you for your very kind attention. Thank you.